Hello and welcome to my podcast. My name is Nika and I am an aspiring scientist from the country of Georgia. Today, I'm deeply honored to welcome Dr. David Managadze, one of the most accomplished scientists in the field. He is a senior principal scientist at DNAnexus, the world's leading cloud platform for bioinformatics, biomedical research, and precision medicine. Dr. Managadze, thank you so much for joining me today. To begin with, could you tell us how do scientists think life first began from non-living chemistry?
Thank you for inviting. My pleasure, first of all, and thank you, Nika, for inviting me to your podcast. Um, okay, so let's. So, how do scientists think life first began? Um, let's talk about the very first steps. Uh, the first steps in how life began were basically creation of the biomolecules. That is. Molecules that are basically building blocks of life, like proteins and RNA and DNA. Mm. Um, and life, unlike the universe, life began not with a big bang, but more like with chemistry bubbling away on the planet, on a really active planet, by the way. Picture Earth 4 billion or so years ago. Oceans are full of raw chemicals. Lightning is polluting the sky. Volcanoes are belching gases, and so on and so on. And one interesting thing is that there's no oxygen, or not much of it, in the atmosphere. Um, so, what could happen on that kind of a planet? In 1950s, it was basically a golden era of biology, right? The DNA has already been discovered, and we were analyzing the structure, protein structure was in works as well, and so on and so on. And so in 1950s, another question that scientists were trying to answer was, is it possible to create biomolecules from non-biomolecules, so to say, simpler chemicals? And so Stanley Miller and Harold Urey at the University of Chicago showed with a wonderfully simple experiment that it is possible to create amino acids, building blocks of proteins, from much simpler non-biological chemicals like ammonia and hydrogen and methane. And so this was a huge breakthrough because before this, scientists thought that it was, well, maybe not scientists, but the kind of a general understanding was that it was impossible to create biomolecules without some divine intervention. And this experiment was a crucial experiment, and they definitely earned their Nobel Prize. Later, scientists, many of them actually ran similar experiments, similar to Miller-Urey experiments, and they detected all kinds of biomolecules in their reaction mixtures. For instance, adenine was detected, which is one of the bases of the DNA, right? Another big step. So now, what we've just demonstrated is that it is possible to create amino acids, which is a precursor, element of proteins, and adenine, which is a part of the DNA. So two of the most important biomolecules as precursors that can be created from very simple chemicals. This is huge. Um, so the next big step was self-replication, because that is a major feature of life, right? So many scientists think this way, and when you think about life, what comes to mind first is that they self-replicate, right? So they create their progeny And that is the major feature. Many scientists think that RNA, a cousin of DNA, if you will, may have been the first molecule able to copy itself, so self-replicate. So it can contain genetic information. The RNA can do that. And thus it is similar to the DNA. On the other hand, RNA can also act as a catalyst, like proteins do. And thus, RNA replaced the protein, so kind of dethroned the protein, because previously it was thought that the proteins were the first molecules of life. That's why the name, the protein, comes from Greek word protos or proteus, which means the first, the primary. So now we think that RNA was the molecule that was the first, the most important biomolecule, because it can do, at least on a certain level, what DNA does, and it can also do what proteins do, the catalysis. Actually, proteins, when the proteins are created, the machinery in the cell is called ribosome, right? So the RNA, the messenger RNA, goes to ribosome, and then ribosome um, kind of reads the RNA and creates the protein molecule, right? Protein chain. But the ribosome itself is an RNA-based machine. That's why it's called ribosome. So a particle that contains ribonucleic acid or RNA. Um, so I think these are the most important things, and this is to our understanding, to scientists' understanding. This is how um, the life began. So once you have all these elements, so you basically started from a non-live material, small molecules, non-biological molecules. So you created the biomolecules, at least the precursors and parts of theirs, and then you you go further. Um, you can catalyze reactions and you can self-replicate. And so once you do that, once you can catalyze chemical reactions as well as replicate the information, you have the basis of life. This is how scientists think life began, at least now. I hope I addressed the question.
Sure. Thank you. Uh, Dr. Managadze, uh, what can genomes tell us about the story of evolution and also our connection to other species?
Um, so by analyzing and comparing genomes of various species, genomics scientists can build tree of life, and you have probably seen it, right? So their first one was built by Charles Darwin himself, right? So there's this one single figure in his book. Uh, he said, I think. Um, and since then, we built a lot of other trees of life. I worked in the field of evolutionary and comparative genomics when I did my postdoctoral studies at NIH, and this is what we did, of course, as well as many other things. So, Tree of Life shows quite well how life diversity has evolved. Uh, that every species is connected and had a common ancestor. Okay, there are lots of similarities with other organisms. Um, and even the ones that we think are very different from us. To make this a little kind of use a popular language and also use examples that would be widely understood, I will bring a few examples. So, for example, about 60% of our protein coding genes have counterparts in bananas. Okay, so bananas are very far from us on the tree of life. So meaning that even plants and humans have a common ancestor if you go like far back, well, far enough, so to say. And if you go even farther, you may find similarities with even simpler organisms. Let's go back up the tree of life and look at the great apes. So with chimpanzees, we share 96 to 99% of genome, depending on the use method. But regardless of the method, it's a very, very high similarity, 96 to 99%. So what this all can tell us is that we're very similar, right? And that is important in terms of evolution. Of course, it is very interesting to understand why are we quite different as well. Um, and so analyzing the functions of genes and their modifications helps us understand that. It helps us understand how genomes evolve and how these changes lead to differences between species.
And Dr. Managadze, there's an iconic picture. I think that's actually the only picture Darwin did uh, the tree.
Yeah.
Yeah.
Yeah,
So
a
that
very simple. It just says A, B, C, I believe, and that is this initial idea that it was genius.
uh, and when we talk about human evolution of humans, let me ask you, how did Homo sapiens become different from other hominids like Neanderthals?
Mm, okay, yeah, that's a good question. So let me start with one kind of a remark. Neanderthals were not dumb, unlike some popular opinion. They made tools, jewelry, like they wore jewelry, and they painted their caves, and so on. So they had their own culture, and they probably, of course, communicate. Not probably, but of course, communicated. But Homo sapiens had a few key, really important genetic differences that may have tipped the balance in our favor. Um, One really famous example is a gene called FOXP2. Um, and so this gene is linked to speech and language. And mutations in FOXP2 in humans cause severe speech problems in humans. Um, uh, so, so, so this suggests that this gene plays a role in the ability to articulate and communicate. Interestingly, the gene of Neanderthals produces the protein that's similar. I mean, it's the same sequence as humans, but um, it's not just the protein sequence. Subtle differences in regulation, like when in space and time this this gene is expressed, where does and when does the protein appear, is important. And so um, we believe that these subtle differences in regulation of this gene helped us develop richer language and much better communication skills. So the chimps, interestingly, also have FOXP2, of course, and so their gene, I mean, their protein sequence is just two amino acids different. There's two amino acids that are different in their sequence. Um, but this is this is clearly enough to cause drastical effects there in their vocal ability. Of course, they can make sounds,
The
but
phenotype.
they cannot. Pardon?
The phenotype.
Yeah, yeah. So um, it really affects their ability to speak, right? So other important genes, FOXP2 is a very famous one and of course very important, but there are other genes as well. For instance, R1, which is HAR1. And so this gene codes an RNA molecule, not a protein. So-called non-coding RNA. And it's important for the cerebral cortex development. Which obviously underlines higher cognition and language and abstract thought and so on and so on, right? So R1's rapid evolution is well studied, and it looks like, and so this suggests that it plays a role in reshaping how our brains are wired, so to say. There's also SRGAP2 gene, which is duplicated in humans, unlike in chimps. And so it may have boosted brain connectivity. And what it does is that when the neuron cells are created, initially they are kind of non-mature, and they start developing dendrites and synapse, the axons and form synapses and so on and so on. And once they become mature, they stop doing that. And this gene, its duplication and kind of increasing in the activity, causes the neurons to be remain in that non-mature state longer, so that they can, this basically causes the neurons to form much more dendrites and axons and synapses. So I think it's a pretty good example of like what differentiates us. I think, long story short, I think it's all brain development and the capacity to think abstract thoughts and also communicate. So generally, these small genetic tweaks, like changing one gene here, changing the regulation of another here. So these little tweaks had huge ripple effects. Stronger social networks, better food resources and finding them, and adaptability. That likely really helped us survive when Neanderthals dwindled.
And Professor and Dr. Managadze, this also influence the macroevolution also influence human genomics. And let
Mhm.
me ask you, what is the importance and role of epigenetics in guiding and governing evolution, and why is it important for understanding both evolution and health?
Mhm. Um, what is about so macroevolution? How does it influence?
The
Yeah, so
macroevolution does influence, but what is the
yeah,
importance and role of epigenetics?
right. So the macroevolution definitely influences human genomics. Just briefly about that, so, we definitely, macroevolution is a very large scale evolutionary changes over long periods of time, right? And definitely, when you study human genomes, and not just human genomes, but many other organisms, we see that there are many methods. Methods probably is not a right word, but many things in the genome that actually influenced these changes. Like, whole genomes were duplicated hundreds of millions of years ago, and that gave evolution kind of a playground to have all these spare copies of genes to experiment with. And that allowed it to create all these new functionalities and so on and so on. As for so it is obviously visible. So macroevolution is not abstract, something that we can talk about, some fossils and stuff. We can look into human genome and other genomes, and um, we can see that the macroevolution left its mark in our genome, all over our DNA. As for the epigenetics and how it guides evolution, a bit of explanation perhaps for our audience. Uh, as we grow and develop, our cells multiply, right, and differentiate. And some of them from the same cell, one cell, we have multiple cells. They differentiate, and some of them become muscle cells, some of them become nerve cells, liver cells, and so on and so on. But they all have the same DNA in their genomes, right? Um, but these cells are physically and functionally very different from each other. This is because not all genes are constantly active at all times and in every cell, right? So rather, their activity, gene activity, is strictly regulated in space and time. Every cell type has only certain set of genes that are turned on and at strictly defined times. Okay, not all the time. Maybe some of them are turned on when the cell was just created, right? And then others maybe later on in the later stages. So genes that are turned on in one type of cell, that is also very important. So um, some of the genes are turned on in one type of cell, and but not in another type. And this is what epigenetic studies, the changes in gene expression that occur without altering the DNA sequence itself. So we now know that epigenetic changes can be inherited from parents to offspring, and it transfers the information without changing the underlying DNA sequence. And that is very important and obviously very interesting to study as well. So as for evolution, epigenetics seems to help evolutionary process. It enables initial adaptations when organisms are in a kind of a new environment, something unexpected happened, so the changes happen. It allows them to adapt much quicker than like evolution would need for genetic changes, right? So evolution acts on much longer um, time stretches. And so if epigenetic adaptations are beneficial, they give evolution time to lock these adaptations in genome. Um, for example, we domesticated wolves, right? And they gradually became more accustomed. And at some point, they just followed us everywhere, and they were not fearful to humans. They started eating our food, such as bread, even fully carnivorous animal eating a starch-based product, the carbohydrate. Um, and so they got adapted to it. Um, this initially probably was done on an epigenetic level. Some regulation has changed. They were, some genes were regulated differently. Perhaps some of the, wolves do have amylase, an enzyme that degrades starch, and but just one copy, and it's not very active. Perhaps using epigenetics, that gene became upregulated, and meaning that it produced much more enzymes so that wolves could eat starch, right? So slowly but surely, these changes became afterwards became locked into the genome. And so how did this happen? We found changes in some of the dog genes involved in behavior and brain development to address this first thing that I mentioned, that, wolves were not fearful and so on and so on. And so dogs have some gene adaptations and that has likely made them more social, less aggressive, generally tolerant to being around humans, and at the end, really love being with the humans, right? And another example is that dogs carry many more copies of the amylase gene, any AMY1 gene, for digesting starch. Uh, what that means is that you may have one copy of a gene, or this is called copy number variation. So genomes sometimes may copy the same gene multiple times so that more proteins can be produced in the same amount of time. So the dogs have that, and they are able to actually eat the carbohydrates much more successfully than wolves. So in other words, so long story short, so what might have started with like a flexible little short-term epigenetic shifts eventually got written into their DNA. So leaving permanent traces that distinguish them now. As for you asked about health as well, right? So um, epigenetics is very important to health because it explains how the same DNA can lead to very different outcomes depending on how genes are switched on or off. Um, a few examples, tumors often, for instance, cancer. So tumors often arise not only from mutations, but from epigenetic silencing of tumor suppressor genes. What that means is that through epigenetics, a gene that is very important for cancer not to develop, that's tumor suppressive gene, that's what they call. So if you silence that gene, for instance, TP53, it is possible that tumor will develop. There's no mutation in that gene, but and it's not like loss of function of that gene, but it's just silence through epigenetics. It could also be that activation of other genes, like growth genes, could happen, and so then cells just start uncontrollably growing and multiplying. That's another thing that scientists study. Metabolic diseases are another example, like obesity and diabetes. Um, they can be caused by various epigenetic events as well. There are studies like that. If you know, like Dutch hunger, winter, it's a
Mhm.
historical event, and it's very well known. Another example is identical twins. They start life with exactly the same DNA, right? But as they grow older, uh, differences in diet, stress, environment, and so on, may cause epigenetic marks to accumulate and be different. And that's why twins are different. One twin might even get cancer, and another one does not. So, yeah, a bit of a long story, but I hope this answered your question about epigenetics.
Thank you. And also, now I'm just reminding myself, uh, when humans evolved, uh, now Homo sapiens do not have uh, ferments to digest cellulose, for instance, and
Mhm.
that's one of the uh, instances of evolution, I think.
Right. Yeah, yeah. So, yeah, there are others too. That's a good question. So sometimes, sometimes evolution does not just um, create a new function. Sometimes it's a loss of function. Um, and so evolution doesn't, I don't like to anthropomorphize the evolution because it's a kind of a mindless process, right? If something works, it works. And so, for instance, what would happen, I think, is that we lost that, but it's okay. Okay, so we continued our existence, and perhaps it was even beneficial in some respects. Another example, for instance, is the some olfactory genes. That is the genes that olfactory receptors, sorry. So the genes that create receptors for smell. Um, so we have many smell genes, so to say. Uh, um, but what happened during the evolution is that many of them just were either damaged because of the mutations, or they were kind of turned off. Um, and essentially, they now are pseudogenes, so-called pseudogenes, meaning that they are there, they look like genes, but they are inactive and they're kind of broken. And therefore, we are not that good at smells, right? It's like unlike dogs that have much more, a much bigger repertoire of these genes, and that's why they have amazing smell. But again, losing the olfactory receptor genes, I guess, was okay for us. It did not really affect us that much.
Um, and Dr. Managadze, how do scientists use these big data to study life today? And also, is DNA really just information?
Um, okay, uh, good question. So now we are touching the subject that I've been working for the past, I don't know, 10-ish years or so, probably more than that. So I'm a bioinformatician. And so for us, bioinformaticians, DNA is literally a code with 4 letters. So it is an information. So it's A, T, G, and C, right? Um, and so that what's interesting there, obviously, is that the volume, the difficulty is in its sheer volume. So human diploid genome is 6.5 or so billion letters. And that's a lot of A's and T's and G's and C's, right? Um, and sequencing a genome is just first step for us. Um, and then a lot of fun starts, so to say. So sequencing just one person's genome creates gigabytes of data, but then you need to process that and understand it, right? And so processing, converting, and analyzing, and so on, generates much more data. Um, now, analyzing a single genome is powerful, right? And we did that in 2000s. We spent like $3 billion on reading the first, and that was amazing, right? Brought a lot of progress to us. But so we've gained a lot of interesting information. We learned a lot. However, nowadays, um, bioinformaticians routinely work with tens of genomes, hundreds of genomes, even thousands of genomes, and I can say hundreds of thousands of genomes at this point. Um, for instance, United Kingdom government initiated a project called UK Biobank, which collected the data of 500,000 individuals. Every one of these 500,000 people filled out a questionnaire and answered questions about lifestyle, medical history, nutritional habits, and so on and so on. So it's very detailed. And so we know these individuals' weight and height and body mass index, blood pressure, lung capacity, bone density, and so on and so on. Medical history, of course, if they had any disease or anything. Um, if they had any medication and so on. Um, I was looking at the questionnaire, and there's even a question like how much cheese you eat. So like cheese intake was also there. So it's very detailed, and this is massive data. We have answers to like thousands of questions from every individual, but we also have whole genome sequences of these individuals. And actually also imaging data of 500,000 people. Imaging, I mean like X-rays and MRIs and CAT scans and all kinds of other scans. And so we are talking about 30 petabytes of data here. It's 30 million gigabytes, right? Approximately. And so, by the way, these data are hosted by DNAnexus, so my company. We host it, and that's how you access this data, or where you access this data. So now, why is this a very good data set, the UK Biobank? So you can connect uh, these individuals, these 500,000 individuals, phenotypes, um, physical appearances, preferences, diseases, all of that, all those thousands of data points, you can connect all of these with genomes, and you can filter this data to find very interesting patterns. Um, for instance, so using these the UK Biobank dataset, researchers found that certain variants of a gene called PCSK9 are found mostly in individuals of African origin. It's kind of prominent there, not much in the European population. And that was interesting because all those data sets before were mostly based on European individuals' genomes. And so this was like the first attempt to include others, very diverse, um, phenotype-wise. So, and so this group of African individuals has dramatically low risk of heart disease and cholesterol-related issues, which is very interesting. The signal was very significant, and therefore scientists created, based on these, they created PCSK9 inhibitor drugs. That's how they call it. So basically a drug that inhibits the activity of this of the protein produced by this gene. And so these drugs lower cholesterol and protect against heart disease. So a new class of life-saving drugs was created based on UK Biobank data. I think it's very important. Another example, so besides genomes, the project also started analyzing thousands of proteins in the samples of these individuals, so blood and urine samples. And so this kind of a sub-project started using AI. Um, so they identified many interesting things. And so, for instance, they identified 4 proteins in the blood that reliably predict dementia up to 15 years before symptoms even appear. So these findings are now being developed into potential early detection tests for dementia, and why it matters. So imagine you go to a lab to do like a simple physical exam, and so they do this finger prick test, get a little drop of blood that could flag dementia years in advance, giving patients and doctors time to act way before symptoms actually appear. Um, so there are many other examples like this. There are already thousands of papers. Last time I checked, it was like 9 or so thousand papers already published based on the UKB data. Um, so I think it's a very important project, and analyzing it is obviously not simple. Not only it is massive by volume, as I said, it's like 30 petabytes, but you also need to perform huge computations to analyze this data. And you're not going to do it in like Excel and Word, right? So, of course, emergence of AI. So first of all, emergence of big data, and now the AI really will help us even more in our quest to glean more information from this data. And for instance, it will be very helpful in analyzing these, UK-based imaging data. not just proteins, right? So I meant like X-ray, MRI, and so on and so on. By the way, UK, United Kingdom, started a new project that is also hosted on DNAnexus. It's called Our Future Health, and this one has 10 times more data. I mean, 10 times more individuals, so 5 million participants in the project. So I definitely expect even more progress and big discoveries from this project. Yeah, so I think these are the examples of how big data um, help scientists and how scientists use it to um, to um, so this is how the scientists use the big data to work in the field of genomics.
That's really interesting. And what role does artificial intelligence play in making sense of genomes and health data?
Um, uh, right. So AI is getting more and more traction in the scientific community um, because of its obvious benefits, right? So a few examples would be like AI algorithms trained on thousands of tumor genomes can spot hidden mutation patterns. So again, the genomics example, I'm starting from genomics. Um, so they can spot mutation patterns. It's not always just one mutation, right? So there's a pattern all over the genome. And so humans are not that good, right? When there's a huge, massive volume of information. And we would not be able to identify those. And AI can do that quite successfully. And so these patterns can point to a possibility of developing cancer, or, you know, it can tell you, hey, there's a possibility of developing a cancer here. Or it can also tell what caused this cancer. It could be like a UV light or smoking or something else. And it can definitely help doctors at some point choose correct treatments that will target the specific mutations. Another good examples are um, um, AlphaFold comes to mind. Perhaps you heard for like 50 years, the scientists struggled to predict how proteins fold. Protein folding is one of the most difficult problems in bioinformatics. Well, it used to be at least. And I've done it too. Like single protein molecule, there was software that we used years ago when I was doing my postdoctoral studies. And it is a very complex task. Lots of physics involved and lots of trials and errors. But this is also very important, crucial for understanding diseases and drug design. And AI just cracked it. It mapped the DeepMind, the company, I believe now it's part of the Google. They created a software called AlphaFold, and so AlphaFold basically mapped the 3D shapes of nearly every known protein. There's a newer generation of AlphaFold, and that one even did protein complexes. So it's just amazing what this software can do. Um, there's also image analysis. AI systems are very good at that, right? So AI systems now can read pathology slides, mammograms, X-ray and MRI images, and they can do that much better than human specialists. They can catch very subtle signals in early stages. And so AI does not really make mistakes. They don't get tired or angry. They don't say, hey, I didn't have my coffee, so I missed the tumor in this image. So um, I think it's going to play, it is already playing very important roles, as I just mentioned, but it's going to play a much, much bigger role. Uh, in making sense of genome and health data, and not only.
That's also a very interesting topic to talk about because artificial intelligence, AI, is almost conquering the world. So that's very
Yeah, it is conquering.
relevant.
Yeah.
Uh, and why is cloud computing important for biology and medicine?
Right. So I did mention about DNAnexus and what we do, well, a little bit at least. Um, but to delve into that a little more, thank you for the question. So cloud computing is like it's like a huge laboratory that anyone in the world can work in. For instance, at DNAnexus, we host many petabytes of data. I mentioned, UKB data, UK biobank in short, and only that is 30 petabytes, right? And 9 to 10,000 researchers, um, probably even more by this time, um, thousands of researchers, let's put it this way, around the world analyze it securely. So it allows flexibility and it is much cheaper. So you don't need like giant servers of your own. And when you need to use cloud servers, you can just spin up a large server, use it for your analysis, and then finish it. Also, it allows accessing data from everywhere. And so that another important thing is that cloud levels the playing field, so to say. A small science group in Georgia, for example, can access the same resources, let's say the same UK biobank data, as, for instance, Oxford and Cambridge can do. Um, and it is great for collaboration. Data sharing in the cloud lets different teams um, across the world work together essentially instantly. So cloud computing is very important for biology and medicine, and I believe that it will become even more important in the near future.
Um, Dr. Managadze, do you think one day we will fully understand the genome? And can our DNA really predict what diseases we might get in the future?
Mm. Um, I think it is too complex for that. Um, Fully understand is a bit of an absolutism, and that's why I'm saying so. So yes, we've read the genome, um, and we know its sequence, but and we may understand it to a certain point, but that's just kind of the surface. The genes are, as I said, turned on and off by epigenetic marks. Then DNA is folded and packaged differently in each cell. After transcription, you produce mRNA. mRNA molecules can be spliced, cut and reassembled into shorter fragments. They can be edited. So cell after this, cell makes proteins from mRNA. Then proteins can get modified, combined into complexes, expressed. I'm sorry, transported to different parts of the cell, and therefore be a part of very different pathways, so on and so on. So they can behave differently depending on the cell type and environment where this cell is, right? So the genome isn't a fixed instruction manual. It's more like a living library, if you will, where the same text is read differently depending on space and time. Understanding all these layers of like genome, the epigenome, um, protein level, and so on and so on. Uh, it could take us quite a bit of time. I would say like a century or more. And even then, the genome will keep surprising us. This is what I think.
That's really interesting, especially nowadays, in this
Yeah.
era of
Um,
genomics.
I'm sorry to interrupt, but I think it's okay to have it that way, um, because for scientists, maybe this is a bit selfish, but um, for scientists, like having some mystery and slowly but surely revealing it is something very important. Please go ahead.
Thank you. Uh, and Dr. Managadze, how does genomics really predict, and also how does genomics help us fight cancer? And how do doctors use a patient's genome in the clinic today?
Mhm. Um, so cancer is it's essentially a genetic disease, right? So it happens when mutations cause cells to grow uncontrollably. Yes, there are some, and I also mentioned there are some cases where epigenetics is involved, um, but still, cancer is a genetic disease. So by sequencing tumors' DNA, and performing genomic analysis, we can know exactly which mutations are causing the cancer. And this is possible today. Um, for example, um, if a lung cancer has a mutation in so-called EGFR gene, um, doctors know a specific drug that will work far better than if you just did a chemotherapy. Okay, so EGFR is a well-known uh, mm, uh, marker for the lung cancer. So um, if you see that, you can find exactly what drug would work for this particular gene and this particular mutation. Uh, this is called precision medicine in general. And in this case, we can also use term precision oncology. And what this emphasizes is that instead of, again, as I said, like just kind of a blanket, chemotherapy, uh, you are using uh, you are using a medication that works exactly for this patient and for that particular set of mutations. I work in this field too. For example, uh, or the largest cancer hospital in the US is my client, and I work with them. Um, they have over 1 million patients worth of data, and we do all kinds of really cool analysis. I do work with others as well, but I think this one is a pretty good example. So they are a leading uh, research center in the precision medicine and precision oncology.
Dr. Managadze, uh, do you think one day babies will have their genomes sequenced at birth? Let's
Um,
talk about uh, some
okay.
babies.
Okay. Um, so I think it is likely. So so today newborns already get a blood test, right? So they get this little heel prick test. The doctors draw a handful of, just a drop of blood basically, and so they analyze a few genetic conditions that are very dangerous. In the future, we may routinely sequence their entire genome. The first genome was $3 billion, but nowadays it costs like full genome sequencing costs just a couple $1,000, and this will probably decrease in the future. And this may become just a routine thing. That would reveal risks for rare diseases, drug sensitivities, and so on. Of course, this also is an ethical challenge. Like, do we want to know everything about a baby's future health? Should parents learn if their child has a risk for a severe disease decades later? Um, these are society level questions, so we will all have to answer. This ventures into some, dark past of eugenics, if you will, or maybe a dystopian sci-fi future. If you know the movie Gattaca, if you have
I've
seen
heard
it,
of it.
something, yeah. So it's a wonderful movie. I definitely recommend it. So yeah, um, I think it is likely. That is the short answer.
And Dr. Managadze, we talked about babies, and let me ask you about the consequences. Is there a danger that people misuse these particular genomics for the wrong reasons? For instance, designer babies.
Mhm. Um, yes, there is a big ethical conundrum about that. Um, but most scientists agree we should focus on preventing serious diseases, not like cosmetic enhancements like eye color and height and so on. Um, but history shows that whenever a technology is possible, someone may and will try it. Um, you may have heard about a Chinese scientist that used the CRISPR method to to edit the genomes of human embryos.
Sure.
So yeah, so it was in 2018, 19. So as a result, twin girls, Lulu and Nana, kind of a Georgian name. So twin girls were born, and there was actually um, there was a third child, but it's kind of less known, so separate parents. But no matter. So these children uh, had so their parents, so father was HIV positive and mother was HIV negative. And so this scientist was allegedly trying to basically prevent HIV to be propagated to these children. And so these children had altered so-called CCR5 gene, and that would allegedly make them resistant to HIV. There's a known mutation, so-called Delta 32, that exists in European population. And if you have that, um, the HIV virus cannot bind to white blood cells.
And
And
then
so
that
this
scientist
was
was imprisoned.
right. Yes, that is correct. So this was the first known case of gene edited babies. Um, And when that happened, this announcement just caused a global outcry. The edits were just not clean. So what I mean is that CRISPR, while it's an amazing technology, it often introduces like so-called off-target mutations. So and these can be potentially very harmful. And uh, this was done without any transparency or ethical approval. And you're right. So this scientist was later sentenced to prison in China. So this remains the most infamous example of how genomic technology can be misused and why strong ethical boundaries are essential. But again, if something exists, a technology, I mean, someone will try it, unfortunately, for bad reasons as well.
That's why scientists and the people who have the power of doing this should be so there should be some ethical considerations for those interventions, I think.
Hmm. Definitely.
And Dr. Managadze, as a scientist, what do you see as the most exciting thing genomics will do for human health in for instance, in the next 20 years?
Um, oh, well, I see a few big things, and but some of them, I've already mentioned this, but so routine genome sequencing probably is one of the more important ones, at least from my perspective. So every patient's genome is sequenced, and then you kind of don't have any secrets anymore. And so then you you can predict things, you can be ready when, let's say, something happens. Another thing, of course, related to this would be, again, the precision medicine that I mentioned. So basically, treatments tailored exactly to an individual patient. Um, and I would say AI-driven everything. So quick and precise diagnostics, drug discovery, and designing completely new classes of medicines, even in months instead of decades, which it does take now. So, and many more. And this is not a science fiction. This is the trajectory we are on. These things will become a reality quite soon.
Uh, From my memory, I think one of the biggest challenges the healthcare system and scientists witnessed in recent years was the COVID-19 pandemic. And let me ask you, what role did genomics play in the COVID-19 pandemic? And could it stop the next one faster?
No, um, I'd say genomics was an undisputed hero of during the COVID-19 pandemic. If you remember, within weeks of the first outbreak, so scientists sequenced the SARS-CoV-2, the coronavirus, um, and uh, and they posted its genome online. It was available for everybody very quickly. Um, so that genome was the basis for the mRNA vaccines by Moderna and Pfizer and others. And so without genomics, those vaccines would not have been ready in such a record time. Um, if you remember later, genome sequencing was also crucial for spotting variants like Delta and Omicron. Um, and genomic labs around the world worked tirelessly, uploaded like millions of viral genomes to databases, and that um, let us track the outbreak almost in real time. Like we would see the news, right? It's like, oh, it's in that country, and oh, that's like Delta, and oh, this one is Omicron. And so this never ever happened before to work through this huge problem, address the pandemic in such a record time. I think genomics was crucial for that process.
And I think that was one of the biggest achievements of science to develop
Absolutely.
mRNA vaccines in such a relatively short period of time. And I think
Hmm.
um, we should appreciate that because
Yeah.
as far as I have read, scientists are working to develop the mRNA vaccine based. They try to develop uh, mRNA based vaccines for HPV, HIV, and other diseases. And I think uh, that's one
Oh
of
yeah.
uh, that's one of the reasons why should why we should trust science and scientists in particular. Dr.
Definitely.
Managadze, thank you so much for sharing your insights and helping us understand the incredible power of genomics and
My
modern
pleasure.
science. It has
Yeah.
been a privilege to learn from you today. And to our listeners, thank you for joining us on this journey through the frontiers of biology, medicine, and technology. I hope you found this conversation as inspiring as we did. Until next time, stay curious and keep exploring science.
My pleasure. Thank you.